All right. Welcome and thank you everyone for joining us for our BRAG meeting today. For those of you who are new to BRAG or not as familiar, the mission of BRAG is to provide a forum of collegial interaction in the research administration community. We do this by facilitating presentations by subject matter experts like Bob today, creating an environment to foster peer-to-peer exchanges, and seeking and sharing best practices. I do have two quick announcements before we jump into Bob's presentation, but I do ask that you guys please ask questions in the chat and please stay muted. If you would like to remain anonymous with your questions, please send them directly to Susan Gardner. And real quick, two announcements. NIH has updated the requirements for other support or current and pending. This was done back in January, so I hope this is not really new news. I won't go into all the details on other support, but there was a fantastic presentation on the subject at the February RAC Forum. If you were not able to attend last month's RAC Forum, you can watch the recording at that web link. Today is also the deadline to submit your application if you would like to join us on the BRAG Steering Committee to help plan and facilitate these sort of meetings. If you have not already done so, and if you are interested in joining the steering committee, please fill out this short Google form. And again, today is the deadline. And that's all my announcements for BRAG. I'm going to hand it over to Daniel Ettlinger to do a quick introduction. And then we'll be on our way.
Thanks. So yeah, I'm going to give a little quick introduction of Dr. Knight. I'm sure it won't do you justice, but I'll give it my best attempt. So Bob Knight is a professor at UC Berkeley with a research program focused on human neuroscience. He received a BS in physics from the Illinois Institute of Technology in 1970, his MD from Northwestern University Medical School in 1974, trained in neurology at UC San Diego from 1976 to 1978, and did his postdoctoral work in human neurophysiology at the Salk Institute for Biological Studies from 1978 to 1980. He was a faculty member in the neurology department at UC Davis from 1980 to 1998, and has been in the psychology department at the Helen Wills Neuroscience Institute at UC Berkeley since 1998. Knight's research focuses on the role of prefrontal cortex in human behavior. He is twice the recipient of the Jacob Javits Neuroscience Investigator Award from the National Institute of Neurological Disorder and Strokes, and an IBM Award in Cognitive Computing and Humboldt Prize in Neurology. He is the founding editor of the Frontiers for Young Minds. And without further ado, I'd like to introduce Dr. Bob Knight and just really express our gratitude for you coming to talk with us.
Well, thank you very much, Daniel, and really thank you for inviting me. And I just want to thank everybody out there for all the wonderful support of the investigators at Berkeley. We wouldn't really have our programs if it wasn't for you. And I don't know who's online, but particularly the people I've been working with, Marina, Kirsten, Jody, Suni, and Jason, who've really facilitated a lot of my work. So with that said, I just want to mention, you can see Helen Wills there. She was a Berkeley undergrad, and she became obviously a spectacular tennis player. She won Wimbledon 8 straight times, and interestingly, she never lost a set in those 8 years. And she married well, and then she nicely left a very large endowment to start our institute. Okay, now with that said, what I'm going to do today is something a little unusual talk for me. I have talks about clinical syndromes, aphasia, cognitive function, emotional problems, and then I have my more mainstream research talk, which is physiology of these processes in humans. And I kind of merged them today. So I'm going to show you examples of patients with neurological problems, and then show you how we approach understanding how the brain actually supports, let's say, language or emotional processing or attention. So with that said, okay, so first, why frontal lobe? Now, this slide, it looks a little gory, but this is reality. You can see the different kind of the damage that affects the frontal lobe. Tumors, strokes, degenerative processes, brain trauma, surgical misadventures, leucotomy in the 70s for schizophrenia, bullets through the brain, dysplasias. There's so many things that affect the frontal lobe from a neurological and neurosurgical perspective. It's really important to understand how this key brain area supports human behavior. Now, you can take this exact slide and just replace the names with other psychiatric disorders. So the frontal lobe up on the left, that's a big tumor, but it's also been implicated in schizophrenia, the orbital frontal cortex, and obsessive compulsive disease, and so on. So if you look at pretty much any one of your major psychiatric disorders, there's something wrong with the frontal lobe in controlling the rest of the brain to produce good, socially appropriate, normal behavior. So I'm going to talk about two areas of the frontal lobe, and the top left is a lateral view of the frontal cortex, and you can see all this here is lateral frontal cortex, all of it. And it's about 35% of the cortical mantle. It's much higher than any species. For instance, non-human primate macaques, it's about 85% bigger. And then if you flip the brain over, the orbital frontal cortex, and as a first pass, you can say the lateral frontal cortex is cold cognition, thinking, planning, attention, language, etc. Whereas the orbital frontal cortex is really involved in social regulation, how you perceive people, how you interact with people, how you respond to emotional events. So what I want to first show you is a patient who unfortunately has bilateral large strokes in his lateral frontal cortex. This is probably why I'm giving you this talk, because when I was a medical student, I was introduced to Alexander Romanovich Luria, the brilliant Russian neuropsychologist. And he basically, based on just observing people with frontal lobe damage, proposed that the frontal lobes were essential for organizing intellectual activity as a whole, programming of the intellectual act, and checking of its performance. Extremely prescient. All of these things have been now confirmed by various modern technology, imaging, EEG, intracranial recording. So this gentleman, what you'll see is when you lose both of your frontal lobes, your lateral frontal lobes, you are disconnected from the world. He looks somnolent, and he just is not able to do anything. It's a bilateral grasp reflex. These are things that you see in babies that reemerge when your frontal lobe is damaged. They grasp. Let me just check some things here. You'll see his lips are going to purse. It's like babies. It's these primitive suck reflexes. They all reemerge. There's a very prominent, say 4+ snout reflex. And he's just under aroused, and that's because the rest of his brain doesn't work. Open your eyes. Open your eyes, sir. Open your eyes. The rest of his brain doesn't work because the frontal lobe is orchestrating function in the rest of the brain. And when you lose it bilaterally, this is what you have. Someone who really is just a bullock and can't do anything. It's a very devastating syndrome. Now, what I'm going to do today is we do research with brain lesions in my lab. We do research with scalp EEG, but I'm going to focus on only one thing, which is intracranial recording. And I'll show you what that means in a minute. The EEG, which you all have, right? You all have electrical activity. It's the summed activity of brain cells in your cortex. And it was discovered in 1929 by a psychiatrist at the University of Jena, Hans Berger. Mainly, he made technical advances so he could record. And he showed quite clearly, this is gray matter. This is white matter. This is just an oscillator giving a time code. And what you could see is these brain signals that you record on the scalp are actually coming from the gray matter. That's 3 to 4 millimeter band of billions of neurons in your cerebral cortex. And there's no signal coming from the white matter. Now, he was able to prove this by looking at surgical patients and putting an electrode on the gray matter and a little needle in the white matter. And he actually, way back then, made this phenomenal discovery. And he was really hoping to use EEG as a tool for psychiatric diagnosis. Now, what we do, unfortunately, about 1% of the population in the United States has epilepsy, and I'm sure someone on this Zoom meeting knows somebody. Now, again, about 80 to 85% of the patients, we can control the epilepsy with medications, but 10 to 15% we can't. And if you do the numbers, that's 400 to 500,000 people. Some of these people, if we can precisely localize where the seizure is starting in the brain, we can surgically remove the tissue and get control of their epilepsy, sometimes complete stoppage or now control with meds. And there's two things we do. We have to unfortunately open the skull, put a big grid of electrodes on their brain. You can see 3 separate patients, and put the skull back on, and they sit in the hospital for 4, 5, 6, 7, 8 days until we get clear localization of their seizure. Most of our cases now, though, we actually do what are called depth electrodes. We do a teeny little hole in the skull, 2 millimeters, and then we robotically target key brain areas that are very likely seizure sources. There's one going into the hippocampus. Here's another one going into orbital frontal cortex. Here's one going in the cingulate. And if you notice, all of them, at least these penetrations go through lateral frontal cortex. So this is giving us access to the actual spatial and precise timing of neural activity in the brain. It's the only technique that can do this. fMRI is great, but fMRI has a 4 to 6 second lag. So 1, 1,000, 2, 1,000, 3, 1,000, 4, 1,000, 5, 1,000. You get the fMRI response. But when I clapped, your auditory cortex, which is right here, was activated in 15 milliseconds. So we really, this technique lets us go into the kinetics of the real-time spatial temporal kinetics of what we do as human beings. Now, one of the things we do is called frequency analysis, first described by Joseph Fourier way back when, and it's really pretty simple. It's a technique that you can take any signal, let's say you're in a jazz club, and this is the sound you're hearing to your ear. You can decompose it into a slower frequency, maybe a bass, maybe an intermediate frequency, a sax, and then a higher frequency, maybe a piano. It's the same principle as taking a white light and breaking it into its constituent signals. And that's what we do with all the data we record. Now, it used to be felt that there was no brain activity that you could record that was over 50 hertz. What is 50 hertz? That's 50 cycles per minute. That's pretty fast, right? Well, it turns out when you put an electrode in the brain, directly in the brain, not on the surface with the EEG, the whole new signal space emerges. And it is very robust, and it goes from 60 to 250 hertz. I mean, think about that. That's 250 oscillations a second. This is the key signal in the brain to track human cognition, because it's also the signal that correlates, for those of you who work with animal researchers, with single unit activity. So it's a really fantastic signal to study human brain function. Now, one other thing is there's a phenomena of coherence that I want you to, so we got this high frequency activity, which is tracking locally, local cortical activity. And then we can look at coherence. And what's coherence? And the best way to describe coherence, I decided, was a playground example. You're going to see 3 kids on a swing. Mom pushes one, dad pushes another mom pushes the third. They're all out of sync, but the kids want to become coherent. So they're swinging at the same speed, and they can look at each other and laugh and whatever they want to do. So here's coherence in the playground. First kid swinging. So there's their sine wave. The second kid is pushed. They're out of sync. The third kid is pushed. All 3 are out of sync. But what those kids will do, they will control their swinging. Until they become coherent. Okay, maybe it took them, 20, 30, 40 seconds. Well, that same phenomena happens in your brain, except it happens in less than 100 milliseconds. So this is another key principle, is that your brain is operating as a distributed network, rapidly moving information back and forth. So if 2 brain areas are in sync, they can communicate. They can communicate. If they're out of sync, they can't. So this is another important principle: coherence, brain networks, coherence. Okay, so now let's dive into a little bit of the data And what I'm going to do again, I'm going to show you a blended talk where I show you clinical examples of people with problems. And then I'm going to show you some brain data from recording intracranially that gives us some insight into how these brain systems work. So most left hemisphere, 98% of males, 92% of women have language in the left hemisphere. And there's two key areas. The front part of the brain right here, it's called Broca's area. That's key for speech output. And then Wernicke's area in the back of the temporal lobe right here. And that's critical for decoding what you're hearing, for understanding what someone's saying to you. And it's language. It could be speaking to you or writing to you or using sign language. It doesn't matter. It's language. Broca's, Wernicke's. So let's take a look at two patients briefly. There's Broca, nice sideburns. This is a patient who has a stroke in Broca's area. What you're going to see, he understands my questions, but he can't output speech. And he makes naming mistakes. So let's just watch a little bit of him.
Let's check your language out now. Can you tell me what happened when you had the stroke?
Yeah, sitting that sorry.
He can't speak. He can't put
do you
out
remember,
normal
was it in
language.
the morning or the afternoon?
TV, no, TV.
11?
Yeah.
11 in the morning?
Yeah.
11 in the morning. Okay, so he's understanding what I'm asking him, but he has a non-fluent output of speech, sometimes just single utterances, but he's trying to communicate the correct content. He knows the question. He's showing me it's 11 a.m. Can you tell me, where were you born?
63, 68.
63, 66.
60.
I see, okay. Where were you born? What city?
Winded.
Where?
Winded. Linwood.
Disordered
Linwood?
language
No. Winded.
output.
Let's just try some simple language things. What's this called?
To be.
What?
To be is to be.
A 2B. It's kind of a phonemic error for toothbrush. He certainly knows what it is.
Because he showed us how it
So
worked.
He is.
How
They
it
know it. What's
actually-
this called?
Key.
Excellent. What's this called?
Key. Key.
Okay, I think that's enough. So you get the feel that this gentleman he understands, but he can't communicate. Here's a gentleman who had a stroke in the back part, in the temporal part of his left hemisphere, and he can't follow anything. And when he speaks, you can't make any sense out of what he's saying. If I asked him to do formal commands, something very simple like, raise your hand or open your mouth that you've heard 10,000 times when you're a baby, he can't do it. So let's just watch him.
How are you feeling today, sir?
Pretty good, yeah, actually. I just not a pest of where I'm going, but I'm matching there, and then I got things moving on vegetables, vegetables, but I'm really not actually, not really in hand. I'm just on there that van.
Are you having any headache? Does your head hurt you at all?
Yeah, it gets where you are on your S. It's not on my Esper in sense, but it's whatever S was Dan that I would not have heard.
So you can see in this gentleman, he's just speaking like I understand him, but I don't, and I'm sure none of you do. He speaks a lot. Unlike the Broca's aphasia patient who's having trouble outputting speech, he can't understand, and he actually acts almost like I understand him, which of course I don't. And I'll show you one more recent case. And by the way, you say all these patients have consented to be used for teaching. So, there's nothing, we're very rigorous about that. This is a couple, really delightful couple that I just saw recently in the Berkeley aphasia program. They're interesting in many ways, but one of them, they're both neurologists. And the gentleman had a stroke in Broca's area, and you're going to see a phenomenally interesting finding, which is that he can't speak, but he can sing. And you'll see in this patient, most people say they can only sing overlearned things like happy birthday, but you're going to see that's not true. And this one patient has led to us putting together a grant to understand the physiology of singing in the brain, because it might have treatment implications. So let's take a look.
Try this, okay? Do you have grandchildren?
No, I have, but near.
Not nearby.
Not nearby.
Yeah.
Broke
Not
his
nearby.
aphasia.
Yeah, so you must have been, let's just do, let's try happy birthday. Okay, ready? With me, ready?
Happy birthday to you. Happy birthday to you. Happy birthday, dear Stephen.
He's one of your
Happy
research
birthday,
subjects. He
dear
can sing, right?
Yeah.
This is an old, he basically can sing and he has,
The
now
melody.
say the words.
No, that chance. Happy
Say,
birthday to.
there's your signature patient.
Mr.
How is that happening, right? I mean, amazing. Now watch what happens. Oh, we don't have the next tape. Actually, I'm sorry, I didn't have the full tape. He actually then, he's a gourmet cook and he's frustrated because he can't order things in stores. And I actually got him to sing orders. And unfortunately, I thought I had the right tape in here. So it's really quite amazing, and it raises issues if you can figure it out. Maybe you can figure out ways to use the ability to sing to improve the communication capacity of people with aphasia, of which there are roughly 500,000 people in the United States. So how do we approach this from a physiological perspective? Work done by a grad student from Berkeley. Here's your brain and fMRI. You show these Broca's area, Wernicke's area. But what you can really do from direct brain recording, we're going to do a verb generation test. Super simple. You say to the person, ball, and you have to generate an action verb. This is used a lot for mapping for pre-surgical. What you're going to see is this high frequency brain response precisely and beautifully tracks language. And I had it happens in 2 seconds. You do the test, less, 1.5 seconds. So I spread the movie out to 10 seconds, and it's just unbelievable what you can do with this technique. Okay, here we go. Say here, ball has to generate kick, Wernicke's area, Broca's area, motor area, say it, refire. The whole thing happened, you can see, in under 2 seconds. This is the only method that you can precisely track cognitive function in humans with millisecond precision. So this was an exciting finding. And because it was so new, I have to tell you, we had a lot of time getting it published. They kept telling us, why don't you just do fMRI? And I was going slightly crazy. But anyway, now it's spread all over the planet. This is a very, very powerful technique many labs are using. So how could you use it for, could we somehow take these signals and turn them into something that would be translational? For instance, let's say you have amyotrophic lateral sclerosis, right, and you can't speak, or some other horrible brainstem disease that you can't speak, but your mind's intact and you're discommunicated. Could we come up with a speech prosthetic device that we could implant over your auditory areas and actually decode your imagined speech? And I'll just show you what we've done. Multiple studies. I just picked out one, and there are 2 people here that did this. Brian Paisley, who has an NSF grant now, but he's moving to Apple. Eddie Chang was a postdoc in the lab. He's now he's the chairman of neurosurgery at UCSF, and he's extremely talented. So the idea, what we did is we took words, we presented them to patients who had these grids over language area, and we recorded their intrabrain EEG, and we came up with models to see if we could decode what part of the sound file each electrode represented. And I'm not going to go into all the details of it, but it turns out what we were trying to do, pretty simple, what we're trying to do is read the notes of the cortex. Just like each piano has a different sound that it produces, each electrode of this complex sound file, let's say you're hearing baseball or tree or whatever, can we decode it? Because if we can, that's the first step in showing we can actually use EEG to extract powerful language information, which is the preamble to now doing imagined speech. And we've done several studies in that area, and I'm not going to really go into it. But we are making progress. And Eddie Chang's lab is doing extremely well also in this area. But let me show you the results. Okay, so what you're going to hear here, the patients heard lots of words, and here's 4 of them. So Waldo, structure, doubt, property. That's what they heard. Then we recorded from their brain, and this is a smaller grid. This is a very large grid. And then we did our reconstruction. Could we take the EEG data and turn it into speech? So first you'll hear
Waldo.
spoken, reconstructed.
Structure. Doubt. Property.
So not bad, right? You can make out all the words, and this was 2012. This paper was published, and we've made a lot of advances, but this was our first major paper. So I thought you'd enjoy hearing that. This tells us that yes, if we, with the proper bioengineering techniques to get the signal out of the brain, and with the things that are happening now with the fusion of neuroscience and bioengineering and computational biology and neuroscience, the future is bright for actually making real progress into neuroprosthetic devices. How about attention? Attention, where language is in the left hemisphere, attention's in the right hemisphere. And here's a patient who's had a right hemisphere stroke. And what you're going to see, he doesn't pay any attention to anything on the left side of space, nothing. My colleague waving a $20 bill, and he immediately orients. And that's because he has a severe problem with paying attention to the left side of space. Here's another gentleman, he's recovered a little bit, and just watch his performance. I'm putting a piece of paper in front of him and asking him to cross out the lines. And he'll cross out the lines on the right side, but not the left side.
Put a cross through every line that you see there.
This is my colleague, Mark Desposito, examining him. You see, he's crossing out all the lines on the right side,
You think you're having
but
trouble
not the
seeing
left
things
side.
on the left side?
Oh, yes. I have to. When I read, I
Classic
have to
neglect of
make
half
sure
of the
I
world.
Go all the way over to the left side to start the line.
And it extends to
I'll
other
flower.
things. So
Flower
if
in
you
a pot.
have him draw a flower, his mental representation only has the right side of the flower. He only draws the right because he doesn't have the mental representation of the left side of the object. It's a very amazing syndrome. Now, and I'll show you a real world example of this. This is for those of you who've been to Milan. This is the Piazza del Duomo, which is a huge piazza. At one end of the piazza is a church. At the other end is a government building. The Milanese, they know all the stores on each side because they've been to them, they've shopped, etc. So they know where they are. So here, this is what happens in patients in Milan who've had a stroke, just like that gentleman, those two gentlemen I just saw you. The doctor simply says, what I want you to do, ma'am, is make believe in your mind's eye that you're on the steps of the church and you're looking at the government building. Tell me what you see in your mind. And since the patient has neglect of the left side of the world, the only things that she reports are the stores on this side when she's looking here, on the right side, nothing on the left side. Now, the patient hasn't moved. She's in her hospital bed, and I say, ma'am, all I want you to do is make believe you're on the government building looking at the Duomo. Tell me what you see. She now reports all the other buildings. So they were there. They're all in her mind, except she couldn't access them. This is just an amazing, amazing syndrome. When you see these patients, you're going, whoa, this is quite amazing. So this is some work by a postdoc, Serge Shapansky, and we were trying to get at the physiology. And these are patients who had a lot. These are 8 patients with these electrodes implanted for epilepsy. And they simply have to do a task. They're being asked to cue to the arrow, press a button whenever something comes up on the right side. Nope, that was on the left side. That's on the right side, press a button. Really simple task called the Starry Night task. That's not attend. So she's either attending to the right side of the world or the left side of the world. And then we switch and have them attend to the left side, not the right side. So what happens? Pretty amazing. This is, remember I mentioned to you this high frequency response that goes up to 250 hertz. If you take an electrode and you're not paying attention to the right side of the world, basically, if you're not paying attention to the right side of the world, you get very little high frequency activity in that electrode. Now I simply switch. I say, okay, now attend to the right side of the field and detect those two blips, those two little boxes. You get a massive increase in high frequency activity. So this tells you this particular method. You can study anything you want. You can study attention. You can study language. And interestingly, if you remember, I mentioned coherence. Not only does the activity in individual electrodes in parietal and frontal cortex massively increase when you're paying attention, the coherence between the two brain areas go up. So the front and the back of the brain become coupled so the information can be transferred back and forth. So you're seeing here, we're actually getting at the physiology, the physiological basis of how your brain allocates attention to the world. And of course, you don't have to go very far to think of not just patients with strokes, but think of all the kids with attention deficit disorder and other things that might be amenable to intervention as we understand the circuitry of these, of how the brain supports attention. We can maybe go in and modulate it, maybe with some extracranial stimulation. Maybe some of your people's grants you're handling are doing brain stimulation. So again, physiology of behavior.
Hey,
I
Bob,
wanted-
we had a quick question come in on communication earlier. For folks whose Broca's and or Wernicke's areas are affected by strokes, are they still able to communicate using American Sign Language or writing?
No, it's a linguistic problem. That's a great question. In fact, my first paper ever, it published in Brain, I think in 1979, I'm getting a little dated there, was actually aphasia in an American Sign Language person. And they had a lesion right in Wernicke's area, and they couldn't understand sign, and they couldn't produce accurate signs. So it really is not a sensory problem. It's a linguistic problem. So if you can't read, you usually can't write. If you can't understand speech, you can't read, etc. So it's a higher level. It's not a sensory level. Yeah, that's a great question though. Now, this is some recent work. I'm not going to go into it. I just want to point out to you, this is Julia Kam, and she was a great postdoc. She's now an assistant professor in Calgary. And she's been studying internal attention. And what's internal attention? Well, it's poorly studied, but it's 50% of your life. And you're doing it all the time. Some of you are doing it right now. You drifted off and you're thinking about what you're going to do tonight. It's internal thought, and it's an exploding new field of research. And I'm not going to go into her studies, but again, using intracranial recording in our different hospital settings with patients, and you can see where the electrodes are, she's been able to make tremendous progress in understanding what happens when you're doing internal thought, what actual brain areas are doing that. And again, very important, because internal thought can be great. It can be relaxing. It could lead to creative thoughts, but it also can have a downside. You can have rumination and worry. So, and there's different patterns of physiology that link to these. Again, understand the physiology, targeting to hopefully fix it. All right, so now the limbic system. So the limbic system is this, it really refers to the brain areas that control emotion. How do you perceive an emotion? And how do you use that emotion to properly have social interaction? And I'm going to show you a little bit of that evidence. The two big areas are the amygdala, which is in your temporal lobe, and the orbitofrontal cortex, which I showed you before, is on the undersurface of the frontal lobe. And these are critical, not for thinking and planning and attention and language. They're critical for how we perceive, extract emotion, and how we are able to do proper social interaction, how we can have theory of mind and understand what other people are, when they're interacting, what they're doing. This very important capacity that develops in kids at about the age of 3 or 4, and of course is not well developed often in kids with, is not well developed in certain kids in autism spectrum disorder, as well as other neurological problems. So let's talk a little bit about this. First, let's talk about the amygdala. And here's some work from Ji Zhang, who was a bioengineer and then did a PhD and worked for me and Jacqueline at UC Irvine. And she was interested in what's actually going on physiologically, when you perceive something emotional, and we've known for years this red area here, which is the amygdala here, is if you damage it, you lose a fear response. So how does that, and we also know that the amount of activation in your amygdala when you see a stimulus, if it's emotional, it'll boost your memory. And you all know that phenomena. You remember many things that have happened to you in your life that were emotional, and you probably don't remember what you had for dinner 2 nights ago, right? But that emotional saliency really captures your memory system, your hippocampal memory dependent system. So she's at MIT now, finishing her postdoc. She's on the job market. We showed the patients, these are the electrodes, that's amygdala electrodes, these are hippocampal electrodes. You can see she got some really fabulous papers out of this. We showed the patients really simple landscapes, which are neutral. Very easy task, just look at the movies. And then we showed the patient movies with fearful faces. And the bottom line, remarkably, it's been known for years that emotion boosts your memory. Everybody knows that, not just the scientists, everybody knows that. Turns out what she was able to show in a series of elegant papers, really quite elegant papers, is that a signal in the amygdala, only when you see something that's emotional, not something that's neutral, sends a signal to your hippocampus and amps up your hippocampal memory storage. So she was actually able to determine the physiological basis of why you remember something emotional. Pretty remarkable work by her. She's really very talented. Now here's a gentleman with orbital frontal damage. Remember I showed you the first movie was the man with lateral frontal damage. He couldn't do anything. He was just a bulk and gone. He couldn't control anything. Here's a gentleman with massive damage to his orbital frontal cortex. That's his MRI scan. He's one of our research subjects. And you're going to see he's smart, but he has no insight. And he's had a very chaotic life because he can't really organize his behavior. He's impulsive. He makes bad decisions. And if you don't know about this syndrome, they're often misdiagnosed as a borderline personality disorder. It's not true. It's from this damage.
Did you notice that you were attracted to a lot of women or just not attracted?
I was attracted, very much so.
To a lot of women?
Yeah.
Hypersexual
I
often.
4 times. I put things in my mouth. Everything's an even number. All my 4s, 8s, or 16s. I count license plates. I count flags.
What's the problem that you're not what you thought you would be prior to the accident? Something is impeding that.
I don't have a whole lot of concentration. I don't like being told what to do. I've had trouble maintaining a job ever since the accident. I am not that perceptive.
Can't read people, no
I
theory of mind.
I'm naive.
You can still take advantage of you on the streets?
I believe people.
So you haven't had any fights? You haven't been arrested in the past 20 years?
Oh yeah, many times.
What for?
Twice on assault against my wife. Although, like I said, I need to replace a door. The only thing that got beat up is the door. And for improperly using somebody else's credit card. And I wanted to open a checking account, bounced $2,000 in checks in a week.
Was that the right thing to do?
No, of course not.
Any other arrests?
I had a DUI, 2 DUIs, and a reckless. One in Hawaii, one in Washington. Seems like there should have been more. I guess not.
Okay, so this gentleman, he has his whole syndrome, which could be diagnosed as a borderline personality disorder, or even some people would say tendency to be a sociopath. He's not a sociopath because he knows after he's done things, he knows it's wrong. He doesn't want to do anything to hurt anybody. He has poor insight, not too bad with my wife, I just broke the door, right? He can't read people, no theory of mind. So there's so many other things we could discuss related to orbital frontal cortex, but I think his IQ is 128. So he's highly intelligent. He passes all cognitive tests, but he can have problems in the free field of life. So I'll just show you a couple things we've been doing because these patients are impulsive, right? And they do things that they don't think. And this is some work by Ignacio Saez, who was a postdoc in the lab. He's now an assistant professor at Mount Sinai in New York, doing very well. And I'm just going to show you how we can begin to study using intracranial recording some of the properties of orbital frontal cortex that, for instance, might be related to impulsivity, or for instance, excess gambling or drug addiction, a whole variety of potential behavioral disorders. And it is a little simple. The task is very simple, basically. You basically can either, a number comes up, and if your number, and you can either take $10 without betting, or if the next number that comes up is above your number, you get $30. So you make a choice, right? And that's the day. So here the person said, I'm going to bet, I'm going to go for the $30, and the number was 6, and sure enough, they won $30. And you can see that this is what patients and people do. They look exactly the same. Basically, if the number is a 1 or a 2, they're going to bet every time because there's a bunch of numbers above it, and they could win $30. If the number is an 8 or 9 or a 10, they're probably not going to bet. So when we record from multiple electrodes, these orbital frontal electrodes, and I'll just show you one of his many results. Ignacio is really quite talented. Patients who lose, and there's 2 ways of losing. You could either lose because you bet on 4 and the number 3 came up, so you regret you bet on 4, or you could lose because you took the $10 and 6 came up. So you can have regret for either one. And Ignacio actually isolated a regret signal in the orbital frontal cortex. And I'll just show you it unfolding. Again, tracking high frequency activity. This is a regret signal. As you get the reveal and you lost or you didn't make the right bet, and you can see all these areas in the orbital frontal cortex are engaged. And I should say, one thing I forgot to mention is, if you also notice, the patient I showed you had acquired OCD. At least half patients with orbital frontal damage have OCD, and that's actually led to treatment attempts of shutting off. They have high hyperactivity in this part of the orbital frontal cortex, and there's several multi-site studies trying to shut off this area to decrease OCD, disabling OCD symptoms. So finally, I'm going to just go back to what I really been so interested in is lateral frontal cortex. And I showed you that gentleman right at the beginning who had severe large lesions. This is a man who came into the clinic, and his presenting complaint was, I've lost my ego. He was a delightful person. He was president of his class at Georgetown, had a girlfriend, was a sailor, was an athlete. And in his last year of law school, had a seizure. This is way back in the late 70s. This is an old movie, and I'm, the examiner, and it's in the hospital waiting room, and I'm the cameraman. So I apologize for the quality. This is the patient. This is his younger brother. Here's his lesion. Basically, he's got a lesion in one frontal lobe that's grown on white matter fibers to the other side. It's not as bad as the other person, so he's not completely gone. But he really has no drive. His emotion is blunted, and he never practiced law. He ended up going to Florida and being a tennis instructor because he was a great tennis player. And then he started having problems because he couldn't, in his mind's eye, remember the score. And he finally ended up back in the Bay selling siding contracts. And that's his brother, who's younger and a successful lawyer. So watch my short interview with him.
You felt your ego was disappearing. Could you tell me a little bit about that?
Yeah, that's right. It meant very little to me, setting up a law practice and getting rolling, even though I had gone 4 years of college, 3 years of law school.
Did you have girlfriends in college, law school?
I did.
How about now?
I don't have any girlfriends now.
No
I
drive.
haven't been looking for any of them.
Yeah. Do you notice your sexual drive is decreased? That's
Definitely.
What kind of problems have you been having if you try to do something You try to get something done. What stands in the way?
Top of the list is memory.
Mhm.
I can't remember things.
You still, to me, seem a little bit casual about it.
It's true, what you're saying. Biff is casual and somewhat jovial and good humored about it all. 4 months ago, we lost my mother.
Mhm.
And people remarked, my sister and my cousin remarked that Biff has not shown any manifestation of grief.
Feel great.
Right, and you know, this is a classic. You need to know that that morning I diagnosed him. He came out of clinic. I got a CT scan. He had this malignant brain tumor. He was told, and his family was told, that this was bad. He was being transferred to UCSF, and unfortunately, the survival is probably a year, 14 months, and he still has this facetious jocularity, very, very sad, and emotional blunting. So this is what can happen. His working memory, his ability to keep memory in his mind was gone. That's why he couldn't play tennis anymore, because he couldn't remember if it was 40-15 or 30-30 or whatever. So let's take a peek at some of the physiology underlying how your frontal lobe is engaged in processing. So this is work done by Matar Haller, and there she is. Matar was a PhD student in Helen Wills, and she did some really elegant intracranial recording with patients having increasingly difficult tasks. So here's a simple task: gender discrimination. Is it a male or a female face? Word repetition. I say tree, you say tree, all the way up to a very hard task, antonym generation. I say hot, you have to say cold. And as you notice, as you go up the difficulty food chain, your reaction time, or how fast it is for you to give a verbal response, goes up. And she had a lot of patients, 16 patients, 8 tasks, 38 recording sessions. The work appeared in a major journal. And the bottom line is pretty simple. There are electrodes that are sensory electrodes. Not so surprised. Each one of these trials, by the way, is a single trial. That's how robust this high frequency response is. And I should have mentioned, it is the targeted response for brain-computer interface, because you can get reliable activity at a single trial, one event. And you can see a auditory task, you have auditory regions, visual task, visual regions. When you actually respond here, this is a fast response, and then this is a slow response. And you can see there's high frequency activity right when you respond, and not so surprisingly, you can see that it is centered in your motor cortices. No big deal, but you perfectly track it. But the interesting electrodes are the prefrontal electrodes. It doesn't matter what task it is, an easy task, a hard task, fast performance on a hard task, slow performance on it doesn't matter. These are all the tasks stacked, and what you can see in every condition, in every one of those 8 experiments, you get something you have to do, and your frontal lobe comes on and it stays on. It's gluing together the perception, what I asked you to do, and the action, what you have to output, basically. The glue, it's the glue of cognition. And again, you can see all these electrodes. These are the electrodes that show this incredible persistent neural activity. There's nothing going on here. You're just in your mind trying to figure out what's the answer, basically. So quite a nice physiological representation. And again, I want to just emphasize, we're not just doing this to figure out how people's brains work. One of the things we really want to do is provide robust physiological understanding that can lead to translational applications.
Hey, Bob, just wanted to give a quick time update. We have about 8 minutes left for our meeting, and I wanted to make sure people had time to send in questions. And I know you wanted to briefly talk about Frontiers for the Young Minds. We did have one question come in on chat regarding going back to Broca and Wernicke. And thinking about the patient who can sing but not talk, do you see, is there any sort of difference in how Broca and Wernicke's aphasia can affect someone that is bilingual?
Well, bilingual, if you're bilingual, you get the same, you lose both languages. What happens as you recover, your primary language will often recover first if you're going to recover. But your language, let's say you speak English and you speak Chinese or Spanish, you'll find the same brain areas in Wernicke's and Broca's area, but they'll be slightly separated maybe by a centimeter in terms of the prime source. They're not exactly overlap, but they're really very close.
So there was also an anonymous question that came in. The question was, do you think CRISPR will help brain disorders in the future?
Well, that's a whole separate topic. Yeah, I think absolutely. It has the potential to do gene editing for lots of devastating diseases like, Huntington's and Parkinson's. Yeah, I think, we're at the very early stages. In humans, it's actually getting the ability to do CRISPR in the brain. It's easier to do it on on crop specimens. But yes, I think the answer is CRISPR is definitely a staggering development in terms of gene editing, and it will have applications to lots of things, but not in the, I don't see it in the immediate environment. It may work for simple systems like retinal genetic disorders, and there's some evidence, but that's pretty simple, and you can get the CRISPR, you can control by injecting right into the retina. You can't easily do that in the brain. Okay, so let me just finish because I was going to show you the frontal cortex for humid, just repeat and watch the red. Frontal's on, it's off, you respond. Same humid, but now you have to do the antonym. You're going to see the frontal cortex come on and you chug away, you chug away, you chug away. What am I going to say? And I'm going to say dry. So you can see this glue of cognition. Now, let me just see here. Should I tell you about, I was going to tell you about lobotomies, but since people may have questions, I think I'll skip these last couple slides and go right to the kid thing.
We may have to book you again, Bob.
There's, okay. Yeah, you could talk to my booking agent. That would be me. So we started a journal called Frontiers for Young Minds in 2014 to have a journal where kids would understand the scientific method. Okay. And it's targeted at kids 8 to 14 because high schools have science clubs. And the journal, it's exploded. We just went over 14 million downloads. We released some Nobel Prize articles that were reviewed by kids. We had 52 million social media views in 6 weeks. We're getting up to like Kim Kardashian. Can you believe it? Neuroscience meets Kim Kardashian. So the journal is basically scientists write an article targeted for 8 to 14-year-olds. And we have 6 different areas: neuroscience, biodiversity, math, astronomy and physics, Earth and its environment, and health. And it could be a new discovery, something that you published in a major journal, like you published it in Journal of Neuroscience or Journal of Paleontology or wherever. You rewrite it so the kids can understand it. Could be a core concept. How do we see color? We've got, the journal's open access. You can go to it and download any article you want for free. There's no publication costs. I managed to get them to, I didn't want someone in a third world country who has a $6,000 research budget have to pay $2,000 to publish an article. So it's all free. And the article is reviewed by a science mentor and a young mind. The mentor is typically a grad student or a postdoc. We've had many of our trainees at Berkeley participate in this, but it's a worldwide endeavor. We have 400 associate editors, untold numbers of mentors. And what they do is go through the article with the kids and have the kids understand what's the idea, what's the hypothesis, what's the experimental design, how do we collect the data, how do we analyze the data, and how do we make it a conclusion in the discussion. So we want them to understand the scientific process. It's really, and it's nice for the kids because the trainees could be a 25-year-old with green streaked hair. They'd much rather interact with, because the first authors of almost all these articles are trainees. The actual authors submitting, and it's a nice, and the mentors are young, and it's just really been a wonderful experience. Every article gets a cartoon. I encourage you to go to the website. So here's one on head injury, here's one on carbon, here's one on stem cells, etc. So we have a full-time cartoonist. Well, they're not full-time, but every article gets a cartoon that's salient to the topic. And it's amazing to see what the kids do. Here's some of the kids' reviews. You'll never get these from adults. Adults are too nice, or maybe not too nice, but here's one 9-year-old saying, it would be helpful if they told us how they took the measurements of brains without actually having to remove the brain. That's pretty interesting. Here's another one. The writer of this article did not make it clear why such an expensive and involved research project to begin with. It seemed like a fruitless task. Ouch, that's a rough review. Here's another one. The point is not clearly expressed. I didn't understand the main scientific question because there were so many details at the beginning. Maybe state what the main question is earlier in the manuscript. I think if authors of grants and papers paid attention to the kids, they'd do a lot better. Here's one of my favorites. This seems important, but the way it is written is so boring, I can't even get to the end. Could the authors maybe sound excited about what they are doing? Another ouch. But it's a really fun journal, and it continues to grow, and we're trying to move it into more and more classroom settings. So that's Frontiers for Young Minds. If you got kids, go on the website, see if they're interested. There's a place for them to sign up to be a reviewer, and away they go. Okay, I think I kind of probably. Okay, so what should I do? We're done.
So does anyone have any other questions? If you do, feel free to unmute and ask Professor Knight directly, or of course you can put it in chat.
I just wanted to say that there were a number of comments. Cece Hyde said that's really interesting, the singing. Yin Kua said it's fascinating. Sarah Gogler said agreed, incredibly fascinating. And Hannah, you said fascinating. And then the questions that have been asked. And Teresa Saunders again said, this is really interesting. Thank you so much for your time, Professor Knight. And the last comment from Teresa Saunders, wow, these kids are smart and funny.
They are smart and funny. And human behavior is really a pretty interesting phenomenon, as you all know. It can go awry, as you all know, but it can be really very insightful and powerful. And it can lead to discoveries like CRISPR at Berkeley. Not too bad.
Yeah. Oh, we had one more question come in.
Sure.
Once an area of the brain has been damaged, is there anything we can do to mitigate the effects of that?
Yes. Well, the first thing is, let's say you have a beginning of a stroke. The first thing you want to do is get to a hospital and get intervention to try to stop the stroke from taking out more damage. Once the damage is gone, what has happened and the tissue is lost, there are various approaches that can help return function. For instance, there's language therapy that can really improve recovery of function. In language, there is, I didn't really talk about memory today. There's things you can do in memory. There's increasing use of brain stimulation, where you put electrodes on this skull, and you take advantage of brain oscillations, and you rhythmically stimulate the brain, and that's been shown to enhance cognition. As I mentioned right at the, I'm involved in a project with Parkinson patients where we're using not brain stimulation to improve motor, but brain stimulation to oscillate their frontal lobe at 4 hertz, and that looks like we're able to improve their loss of cognition as that disease progresses. So there's lots of things going on out there. For sure, there's a whole world of brain-computer interface that we really just alluded to briefly, that is being developed for treatment of speech deficits, but also for neuropsychiatric deficits, closed loop stimulation to treat depression. So it's a pretty exciting time, because what's happened, it's not just cell biology anymore. What's happened is a fusion of neuroscience, engineering, and computation. And when you get those three together, that's a powerful nexus, and great things are developing in the field.
Thank you. We had one more question come in. So this person writes, I know Broca's and Wernicke's areas get a lot of attention in regards to language, but can you speak to the impact of the insula?
Yeah. Oh, okay. Thank you very much. We're about to submit two big papers on insula recording. The insula has the back part of the insula is more involved with internal body homeostasis, like heart rate, things that are really involved in controlling, things that are very primitive parts of your daily bodily function. The front part of the insula is really fascinating because it turns out it's linked to reward processing and positive. It's really, I'm glad you asked this question because I took out the insula slide when I was talking about the limbic system because it's now been inserted. It's part of your emotional control. It allows you to gauge positive and negative reward and guide future behavior. So I think you're going to see an explosion of interest in the insula. One paper is coming out from a postdoc in my lab on memory in the insula, and the other one on reward and learning and positive and negative emotion is again from one of our grad students who just finished and now is started a postdoc over at UCSF. So very fascinating, largely underexplored area, but the thing is you can get to it quite nicely with these intracranial electrodes. And I want to mention one thing. We don't put the electrodes where we want to put them to do an experiment. We put the electrodes, all the electrode location insertion is determined by the clinical epileptologist. They decide where the electrodes are. We piggyback our research in the epilepsy ICU onto those patients. I want to be very clear about that.
All right. Well, thank you so much. I know we're over time, and I just want to give a round of applause to Professor Knight for taking time out of his day to come talk to us. We really appreciate it, and this has been a very fascinating talk. To our audience, our next meeting is May 24th, and we're going to be hearing about the IDC negotiation process at UC Berkeley. I put some links in the chat for you guys to our BRAG website, to join the BRAG listserv, Google form to join the steering committee, and a brief survey if you have time. We'll be following up with a thank you email and the survey link via email if you are already gone or don't have time to complete the survey at this time.
Well, thank you again, and thank you for all the work you do to support science. It's really a pleasure to be at Berkeley and to have such great support staff and great trainees and grad students and postdocs. It makes your life a lot more, I don't know what the right word is, enjoyable and meaningful and productive. So thank you very much.
Thank you. And thank you everyone for attending, and we will see you guys on May 24th.
Bye-bye.
Thanks, Professor Knight. Appreciate you.
You're very welcome, Daniel.